tell. And Marnie, we decided, like, you looked at the way I was doing the bumper, we were on board with the okay, I'll go through and finish those up this weekend and just get all
Yeah.
the old seminars up so that way we can have that.
Finish that up. Great. And I can get Michael to post those when you're ready too.
Oh, that'd be good. I forgot he was on the YouTube channel. As we, like the PR meeting thing yesterday went really well as well. So I think that was good. It seems
That's
promising.
great. Yeah, I'll be anxious to hear more.
Michael, I'm trying to find people, like we need to like raise the profile of the center. So we've been talking to like hiring an external PR person.
Oh, that's good.
There's a Duke alum who now has a boutique like PR company and for biotech, and it was like, if you could help us, that would be great. Okay, good. People are starting to join.
Yeah.
Okay.
And I sent you the closing slide.
Yes, I have. And so I just, Michael, are you on board? I guess I'll introduce, like, sorry, I just realized that the bio we have isn't like, it's like more about your group. Is that okay? That's okay.
That doesn't make yeah, it's fine. Don't need to give
You're like, no, you must tell my degrees. But
yeah,
like,
yeah, that's fine.
sorry, I just realized I'm like, I think it sets the tone for what you're going to talk about.
That's fine.
Thanks. Sorry, I just taught and just ran back from teaching, and I'm like, I feel like I'm like not as on top
Yeah,
of
I mean, my class ended 10 minutes ago, so
Okay, good. Yeah. I had someone like trying to ask questions afterwards, and I'm like, I'd love to help you, but I've really got to go.
I'm don't worry about it.
Okay. You want me to start now?
Let's do it. Yep.
Okay.
Let's give another like 30 seconds for everybody to join, but thanks everybody for coming today. We're really excited. We'll give it about maybe 30 seconds, another minute just to get everybody else in the room. Okay, so thank you guys for joining our seminar. This is the Computational and Digital Health Innovation Seminar Series. This is going to be our third one of the semester. We have one more left this semester before we kind of close out the year. As hopefully you've noticed we've we're kind of oscillating between a Duke speaker so you can hear more about what's going on at Duke, and then an external speaker the following seminar. So next seminar, definitely join us back. We have Yang Yu coming to talk to us from Singapore. He is focused on machine learning and applications in health. So he'll be a really great speaker on that end. Right now, we have our last Duke speaker of the semester, so we're really excited to have Dr. Michael Rubinstein joining us. I'll tell you a little bit about his lab, where the research in the Rubinstein group is in the field of polymer theory and computer simulations. The unique properties of these polymeric systems are due to the size, topology, and interactions of the molecules that they're made of. And the goal of his group is to really understand the properties of various polymeric systems and to design new systems with even more interesting and useful properties. Their approach is based on building and solving simple molecular models of different polymeric systems, and the models that they're developing are simple enough to be solved either analytically or numerically, and contain the main features leading to unique properties of real polymers. Computer simulations of their models serve as an important bridge between the analytical calculations and experiments. He's going to tell us today, he's going to be covering a talk called The Loops of Life. He is the Alexander S. Vesic Distinguished Professor of both chemistry and physics here at Duke, as well as a professor of biomedical engineering. We're really excited to welcome you here and thank you for joining us, and I'll turn it over to you. And I guess one comment beforehand is just if you guys have questions during the talk, please raise your hand, enter it into chat, and we'll turn on and jump on. I'm going to turn off my video for this, but at the same time, we're here and we'd love to have an interactive discussion, and we'll turn video back on as soon as anyone has questions. And with that, I'll turn it over to you. Thank you so much.
Thank you, Amanda. So the work I will be talking about today was done by Brian Chen, who was a student here, now the postdoc here at Duke. And the outline, I'll start from introduction to nuclear physics. Then I'll talk about probability of a contact and introduce this idea of loop extrusion. And finally discuss that how you can, if you're active, you can induce some kind of compaction of material. That's what happens, we believe, inside the nuclei. So as Amanda said, it's very hard to give seminars without seeing the audience. So I really would love to have interruptions and questions. So if you either raise your hand or type something in chat and ask Amanda to monitor it so we can have some kind of a discussion rather than me speaking to the screen. So starting from nuclear physics 101, nucleus is the part of the cell containing its most important cargo, the DNA, with genetic information stored in sequence of bases. And to give some numbers behind the statement, nucleus is like a huge library. It contains on the order of 20,000 books, which are genes. Each gene is the sizes between 200 base pairs and 2.5 megabase, and there are 100 times more non-coding instructions that are in DNA. The way it's organized is there are 46 chromosomes in our genome that has the length. Each chromosome is between 50 megabase pairs and 250 megabase pairs, and they are segregated during interphase into territories, which we call like 46 bookshelves. And here you can see they're visualized in a separated way by different colors. And upon differentiations, each chromosome consists of euchromatin, which are regions of loosely packed DNA proteins. It's like something like a reading room. And then heterochromatin with tighter packing of DNA, which you can think about like archives. So the problem we are trying to explain is the genetic information stored in these astronomically long linear polymers, make it very, very challenging to read and access it and read it, which is called transcription. So to give you an idea, it's like a huge, very, very, very long telegraph tape that contains very important information, but it's extremely hard to find where it's stored, which information is stored where. And this plot describes the lengths of genomes of different organisms. And to give you a scale idea, you can each base pair is an order of angstroms, but if you increase its size by 10 to the 7th, so you can think of each base pair as something we can, smaller scale we can visualize as a millimeter, then kilobase pair would be a meter, a megabase pair would be a kilometer, which is size of genome of a bacteria. And then our own genome will be size of the Earth. And then some of the organisms, like amoeba, has genomes of the order of distance to the moon. So think of something, information stored on a scale of a millimeter, and this huge tape is size of the Earth or distance to the moon, which is really enormous. So the main problem is how to pack these thousands of kilometers of tape without getting it all entangled, because entanglements create major problems for cell division. And also, besides entanglements, there is going to be a very special way of packing to have efficient epigenetic regulation, which is really crucial for cell function. Different faraway parts, which are called loci, need to be able to frequently encounter each other to control the process. So to give you an idea what this transcription regulation means, you have instructions, which are regulatory elements like enhancers or silencers, for reading a given gene, a given book, are typically located about 100 kilobase pairs. So now scales will be 100 meters away from along the DNA from the cover of this book, which is called promoter. And so to start transcriptions, they have to encounter, so this enhancer has to encounter promoter and activate RNA polymerase. Or to silence it, or to prevent transcription, you have to have silencer. Just like a cartoon showing here, you have to have enhancer getting close to promoter, and this way, if it would start RNA polymerase, or you need to have silencer that blocks transcription. The problem is that these loops here, the distance between promoter and silencer, or promoter and enhancer, are hundreds of kilobases, which in our scales would be like hundreds of meters away. So for them to frequently encounter, you need to have the probability of contact between promoter and enhancer as to decay with this distance between them, which we call here S, as some very small exponent gamma. Another problem is that how to pack this the strands in such a way to prevent enhancer and silencer from activating or shutting off neighboring off-target genes. So it has to be able to control the gene that it wants to contact and not control somebody else, which is could be nearby. If these loops are very long, it's very hard to imagine how you pack it in such a complicated way. So this is the problem we are trying to solve. How does chromatin is packed so enhancers and promoters meet often, or silencer promoter meet often, and don't do it off target. Also, how not entangle, which will make the whole dynamics there very, very difficult. Any questions on this at this point?
Michael, can I ask a quick question?
Go ahead.
This contact, is it like some sort of electrostatic or?
No, I think it's binding. It's binding. You see here, you can have enhancer, and there's other protein that is allowed it to come close and get binding. There's some kind of weak interactions. You can think about them of some specific interaction. It's not just electrostatics, because electrostatics. Well, I mean all bonds in life have electrostatic basis, even van der Waals or anything else. But it is more special bonding. But the problem is that inside the nucleus, inside water, at our concentration of salt inside our body, electrostatic is screened on scales of nanometer. So, and the scales we're talking about are much, much, much larger. So electrostatic interaction would not be a long range at all. So you cannot really account for electrostatics to see those. If they were charged and attracted to each other, it would not work. Everything is completely screened due to sodium and other ions that are floating around in medium everywhere in cells, extracellular media inside the nucleus. Okay, other questions?
Thank you.
Okay, so we're going to talk about contact probability. And to introduce that, I have to introduce the concept of fractals. So fractals are beautiful creatures, but I'll be talking about science of fractals, which is based on something we call self-similarity. And self-similarity means that if you take an object which is fractal and you blow it up, magnify it, a magnified version looks the same as the object itself. And the way the mass and size are related in pop magnifications is called fractal dimensions. So if you take an object which is a section of an object which is proportional to size r, say twice as large or 4 times as small, mass of that section would be proportional to this r to exponent d, and this d is called fractal dimension. We are more familiar with regular dimensions. For example, for solid three-dimensional object like a solid ball, this d is 3, so mass is proportional to size cubed. A piece of paper, d is 2. For a wire or a line, it is d is 1, so mass is proportional to length of wire. But fractal objects do not necessarily have this integer simple dimensions. And for example, something called Koch curve that I'm showing here, this fractal dimension is a ratio of log 4 over log 3, which is 1.26. It's a rational number. And there are multiple different regular fractals, like Sierpinski gasket that is shown here, where d is 1.59 is log 3 over log 2. And just to illustrate what self-similarity means, you can zoom in and keep zooming in on this fractal, and it's forever going to looks the same as the whole object. You can look any smaller section of it looks the same as the whole object, and you can look at three-dimensional structures like this, and they're also self-similar different actions. If you look at it this way. Okay, so what we're going to be looking is polymeric fractals, which we think chromatin is. And to define a polymeric fractal, you can think about the contour length of a polymer, which we're going to call S, and we are going to be measuring it in bases or kilobases for DNA. has a size R, and the relation between S and R has this fractal dimensionality D. And if you have here, for example, a section I'm showing of this polymer, the volume pervaded by this section, or the section is inside this volume, proportional to R cubed in 3 dimensions. And so this volume is proportional to number of base pairs to the power 3 over D, because it's cube divided 3 divided by D. 3 is dimensionality of space, and D is fractal dimensionality of the particular object. The contact probability of those green beans or green balls or sides to be close to each other with a distance where they can interact and absorb or bond. So there has to be some very small interaction of volume V. Probability of it is the ratio of the interaction of volume V over the whole volume R cube, assuming that they are completely randomly placed there and no long-range interactions. So probability of a contact is proportional to this 1 over volume, or S to the power minus 3 over D. So what we call the exponent gamma, which is what we're trying to make it very, very small, is equal to space dimension divided by fractal dimension. So the more compact object, the larger fractal dimension, the smaller is this exponent gamma D. So gamma is proportional to 1 over D, so equal to 3 over D. So just to give you an example, as I said, the higher D, the smaller is the gamma, and for simplest ideal polymers that are like random walks, fractal dimension D is 2. It's like mean square displacements proportional to the length, and it corresponds to fractal dimensionality smaller than space dimension, meaning that those random ideal linear polymers can, in dense system, heavily overlap. And this contact probability decays very, very quickly with exponent minus 3/2. So I'm showing here a logarithm of contact probability versus logarithm of the genomic distance, and it decays as 1 of, well, S to the power minus 3/2. So the slope of this line on log-log scale is minus 3/2. If you have some other fractal dimensionality, for example, we showed about a decade ago that if you take ring polymers and make them very, very dense, they have fractal dimensionality. They cannot interpenetrate very deeply, so they just stay with partial overlap. Their fractal dimensionality is 3, and the exponent here would be minus 3 over 3, which is minus 1. So we get contact probability proportional to S to the power minus 1. If you're able to make even objects with even higher fractal dimensionality than 3, which is very difficult to imagine, like fractal dimensionality of 4 for this random lattice animals, randomly branched structures, then the contact probability would decay with exponent minus 3/4. So this is the range we'll be talking about between very slow decay with exponent minus 3/4 and very steep decay with exponent minus 3/2. Any questions of what I mean by contact probability and how it relates to the structure and fractal dimensionality? Okay, so I'll be continuing. So we're talking about packing of DNA and proteins in chromosomes, and the way what we know for sure is that on the smaller scales, they form nucleosomes, which consist of about 10 nanometer histone core, which is wrapped around by about 150 base pairs of DNA, and then there is about 50 base pairs of a spacer. So you have about 1.5 cycle of wrapping around of DNA around the histone core, and then you have another section. So it's about 200 base pairs per nucleosome. And on a larger scale than that, people are not really sure how DNA is packed in chromosomes in interface, for example. There are different models, but it's not well understood. So I'll be trying to explain what happens from our point of view on those intermediate scales. And the way people were able to start measuring it about 15 years ago was a technique called high C chromosome conformational capture, where they were binding together or cross-linking two nearby strands of DNA inside the chromosome, just randomly, whatever happens to be nearby were cross-linked. Then the sections near this were cut with distinction enzyme. They filled the ends and marked them with biotin, and then they ligate them so they form connections. And when they purify and fall down with biotin, what they get is sections that of DNA that happen to be nearby, now connected. Then they can sequence them, and then they can find what the probability, how often they find a particular two sections that happen to be nearby. So this is the technique called Hi-C, and they were able to construct the probability matrix, which shows on X and on the horizontal and vertical is the location of this particular base pairs, and then the brightness tells you how often they happen to be next to each other. So they were cross-linked together and then sequenced together. So the first thing one can get is just to calculate how this contact probability, you see along diagonal is a very high probability of contact because those sections are nearby. And moving away from diagonal, you're talking about contact probability between sections that are further apart along genomic distance. And so I can average everything along with a particular distance from diagonal. That gives you the probability distribution as a function of genomic distance, which is what I'm showing here as a very schematic way. What was observed, and I'm showing some data on next to this plot. So left is the schematic representation, right is the actual data. What is observed is up to about 35 kilobase pairs. This contact probability decays with exponent bigger than 1, something between 1 and 1.5. Here, for example, the slope here is minus 1.1. Then between 35 kilobase pairs and 400 kilobase pairs, the slope is much smaller. It's smaller than 1. It's about, on this plot, about minus 0.76. And then beyond 400 kilobase pairs in megabase region, the dependence is again steeper, and it's again above 1. Something like here, an example shows about 1.08, or can be even larger. So this decay exponent is bigger than 1. We can try to understand because that corresponds to dimensionality smaller than 3. And for example, there are models of packing of rings that in simulations showed, this is data showing here is a simulation of dense packing of rings that try to simulate nucleus that have this contact probability on a log scale with exponent close to 1.1. 1.2, which is consistent with this region with slope of steeper than 1. But there is no simple explanation for the exponents decaying much weaker, which is what you really need or want to be able to have a very high probability of contact between promoters and enhancers or silencers, and you also want to avoid off-target contact. So this requires very high fractal dimensionality bigger than 3, which is higher than fractal dimensionality of space. So what we want to understand is how is it possible, for example, for this 0.75, fractal dimensionality has to be equal to 4, because it's 3 over gamma is your fractal dimensionality in this region 2. Any questions so far what we are trying to answer? Okay. So looking into more detail into this map, which is what each brightness of this map, this is for chromatin 14, corresponds to the probability of contact between loci I and loci J along the chromatin. And so this is how it's represented when probability of I and J are next to each other. It's like a treasure map. It tells us how our genome is packed, but it's only tells it in terms of contact probability. It doesn't tell you how it looks in 3 dimensions. So we have to reconstruct 3-dimensional structure from these contact probabilities, or try to understand the physical way of how it does it, how it forms it. So there are 2 interesting features in this map. One of them is this rectangles you can see by eye, which I showed here like in green, which are off-diagonal checkerboard rectangles. And then along diagonal, there are these squares. So these off-diagonal rectangles are called compartments. The typical width in these maps is between 40 kilobase pairs and 3 megabase pairs. And they correspond to association between non-consecutive sections because they're off diagonal. And they are analogous to what people, if polymer physics, observe where they have copolymers that have some kind of microphase separation. So what people identify in our chromosome maps is there are 6 types of groups, A1, A2, and B1 through B4, that have higher correlations to being next to each other, and they have some distinct genetic marks that so they like to stick to each other a little bit more than with other groups. So that's the explanation for this for these rectangles. Then there are squares along the diagonal that are called topologically associated domains. And those squares have size between 20 kilobase pairs and 3 megabase pairs. And they correspond to high contact probability of something which is continuous sections of the chromatin, something that nearby. And genes within these domains are often correlated. And the model that was proposed about 15 years ago to describe 10 years ago to describe this region is called loop extraction model, which is what I'm going to describe right now. This was proposed by Marco and Mirny and Lieberman Alden. So what they're saying, there's some kind of proteins, for example, cohesins that bind to chromatin or to DNA, and they extrude loops through these groups, and they keep extruding those loops until they meet a group that we're going to call stop sign. It's a special section of DNA that has a protein bound to it. And so there's a maximum size of a loop that corresponds to a particular region of DNA to be extruded. And then they unbind and then bind at some other place. And this is how this model of forming those TADs. So these TADs, the corners of the TADs correspond to those stop signs. And the section inside the TADs are corresponding to this loop size. And this loop extrusion was observed in vitro when the DNA was attached to, in this case with lambda phage, was attached by 2 ends by avidin to the glass. There was a flow turned on, and then they added cohesin and ATP. And you can see here that the DNA in the flow formed this arch, but then when cohesin was bound to it, it was moving upstream and forming this two double-stranded loops next to each other, which looks like this thick section. And within about half a minute, it went all the way across this lambda phage. So that was observed in vitro. This extrusion was never experimentally observed directly in vivo. Any questions about the model? Okay, so we are going to be using, and this is what Brian developed, is this simulation model and theoretical model to imitate this extrusion. So you can think about the cohesin or some other, but say cohesin would be our example. It binds onto this polymer of beads, and each of these beads represent about a kilobase of DNA. We're doing cross-grain description, and then it starts extruding. So the two arms of this cohesin are walking away from each other with some velocity Vx, with certain, which is we're going to be quantifying the number of base pairs, the kilobase pairs per unit time. And we assume that these binding sites are located uniformly. In principle, any site could become a bind site. So there's binding probability proportional to length of a section. And in a steady state, the number of bound cohesin is constant because some of them are bind, some are unbinded. This is a dynamic equilibrium in interface state. And this is the model. The model is just you have cohesin binds, extrudes, and then either meets a stop sign or doesn't, and then it unbinds with some probability that corresponds to its lifetime. So the two main parameters, one of them is processivity, lambda, which is how large a loop on average and unimpeded cohesion is going to extrude before it on average unbinds. And the second parameter is this, how dense are those cohesions located that are currently extruded located along the DNA, or how far apart they are. So it's average distance between bound cohesions. Those are the two main parameters of the model. And we'll tell you how we're going to extract it from experiments. Any questions about the model? Okay, so this model can be easily simulated, or we can do analytical calculations. And the first thing we calculated was the full extrusion probability. So it's a probability of two stop signs meeting and the loop becoming the maximum possible loop in a given cohesion. So another parameter here is this capital N, which is the length of a strand between two stop signs, the maximum loop size. And here we are plotting this probability of those two stop signs being next to each other when the cohesions get trapped and stops extruding, when both legs of a cohesion get to two stop signs, divided by lambda, which is processivity, which is the average loop size that they extrude before they unbind. And what you see here is there's function that we calculate analytically, and it gives you an equation for it. It has a peak. It's non-monotonic, and the way to understand it, if you have very short this topological associated domain, or those two stop signs very close to each other, it's very high. It's say close compared to how long a loop can be extruded by a given cohesion, how long is that lives on the DNA before it unbinds. Then with very high probability, this cohesion is going to reach the ends, but the number of those cohesions per the section will be low. So that's why this initial growth of probability of meeting, if the section is very short, probability of binding is very low, and the two stop signs will not be next to each other because very unlikely cohesion is going to bind. On the other hand, if these two stops are very far apart, then the probability of cohesion bound to it is very high, but it's going to unbind before it goes all the way to the end. So you have this exponential decaying probability for very long TED sizes. And that's why this function is non-monotonic. And then we can compare this with experiment, which was done by chromatin interaction analysis with paired and TED, called chia-PET sequencing. And you can see that this functional form agrees with experimental observation. And then from the fit of our experiment, our simulation to experiment, or analytic expression to experiments, we can obtain the fitting parameters, which is our two parameters, the processivity and separation. And they seem to be not very far from each other. Processivity is about 200 kilobase pairs or 190, and separation is about 160. Now we can, using these parameters, we can predict the full map. For choosing specific location of stop signs, we can reconstruct the map, and we're comparing our simulation to experiment and the agreement. So here I show in greens where we put the stop signs corresponding to what was allocation on a particular chromosome, and the correlation was about 0.9. So it was very good agreement with very, very simple model. In fact, we can go beyond that and look not just on the whole map, but specifically along the edges of TED. So here you can see those two corners of TED, which is what correspond to this function here. But we also can look along the edges, which corresponds to the probability of any of the loci along DNA to meet with one of the stop signs, and look at our predictions in comparison with the experiment. And so this is the contact of an anchor or stop sign with the locus located at I plus S. I is location of an anchor, and I plus S location of any loci. And the contact probability that we predict is strongly non-monotonic. It now looks like that. And the data, experimental data, seems to match very closely to this functional form. And we didn't use any additional, we used the same parameters as we got from the chia-PET, 190 kilobase pairs for lambda. 160 for the separation D. And we had to use another function that we didn't have before, was the contact probability distribution with two regions, regions steeper than 1, up to 40 kilobase pairs with exponent minus 1.1, and region with 0.76 at larger scale. So this one, for this calculation, this function was taken as known from experiment. But given that function and those two parameters, we can completely reproduce all of those edges. And in fact, you can look at other edges and they all fit very well with no additional fitting parameter. So this way, we kind of we're happy that we can understand the contact probabilities, details of the map. What we didn't quite understand is how to get this function, where this function comes from, and what creates this very high contact probability of very slow decay with exponent 0.76. So what we want to understand, this experimental function that I was telling you about with the k was 1.1 first and then 0.76. We want to understand where it comes from. And the hint was that if you deplete cohesins, then this region, which has a slower slope than that has a slope minus 0.76, in normal cohesion with a normal action bound, if you deplete them, this distribution becomes, contact probability becomes stronger, decaying, and the function becomes, probability of contact becomes lower. So you need to have cohesion bound and not depleted to get this anomalous exponent with corresponding factor dimensionality of 4. So it gives a hint that cohesins, the action is very important. Any questions so far? I don't want to keep on talking without you asking questions. Okay, so in the second part of my talk, I'm going to try to explain the reason for this lower gamma 2 or higher factor dimensionality of 4, this intermediate regime about 100 kilobase pairs, which has to do with dynamics of this extrusion. So to do it, first have to explain what is the dynamics without extrusion. So if you have some kind of a polymer that is a passive polymer, and look at a section with S loci. If polymer is completely passive and completely random, it mean square size proportional to S. It has factor dimensionality of 2, and its mean square displacement as a function of time is proportional to square root of time. So it moves. Normal diffusion, you have mean square displacement is proportional to time, but since these beads are attached to other beads, as time goes on, they're learning they're part of a larger and larger polymer. The effective diffusion coefficient slows down. At least there's something called the subdiffusive motion, where mean square displacement loci, which proportional to squares of the size of a loci, and and it depends on chemical size or the genomic size. So mean square displacement is proportional to S, and it's proportional to square root of T. So that gives us the properties of just completely passive, thermally moving polymers, like the behavior in polymer solutions, polymer melts when they strongly overlap without any dynamic interactions. And that kind of agrees with what people observe with mean square displacement of sections. You can see here, mean square displacement is proportional to time to the power of ½ Now what we're going to discuss is what happens if you add extrusion. So you have now add dynamics of loop extrusion and see how this whole story changes. So now we have loops that have cohesion binding to this section and actively extruding a loop. And this chemical size of a loop grows linearly with time with this velocity V extrusion that you quantify in number of kilobase pairs or base pairs per second. And so you can plot genomic lengths on the vertical scale as a function of time, and extrusion here shown on log scale as the black line. So the genomic size of extruded loop grows linearly with time. But as I showed previously, relaxation of the section grows the square root of time. And so that is slower than extrusion. So that means that sections that are short sections relax faster than they get extruded, but large sections relax slower than they get extruded. So there's some kind of a crossover here occurring that we're going to call relaxation strand, and sections smaller than this G min, genomic distance of this crossover, are able to relax before they get extruded, but sections larger than G min don't have time to relax, and they get extruded, and they are not relaxed. They're actually getting extruded more and more into the same volume. So you get a lot of compaction. You get this cohesion push more and more of loops in the region, and these regions of loops are not able to relax. So so you can see that the largest section that get extruded is typical value of lambda, which is our processivity, and the section that gets able to relax during this time is the geometric mean of processivity and this minimum relaxation section. So only that loop is able to relax by the time cohesion unbinds. The larger scales are not able to relax, and the whole thing gets dumped into the same volume. So now I want to understand what is the structure of this volume given this cohesion dumps a lot of loops, and the loops don't have time to relax. So the result of this compaction, what we are going to be showing you, you get fractal dimensionality of 4 because of this activity. So the mean square size of a section which is relaxed is proportional to the size of a section, mean square size proportional to the genomic size. So you have mean square size of the extrusion section, or distance from a cohesive that are shorter than the relaxation blob. They relaxed, and their size is proportional to the mean square size proportional to the genomics distance. This is just statistics for dimensionality of 2. Mass is proportional to square of size. But on larger sections, they don't have time to relax. They move subdiffusively, following this anomalous subdiffusive motion with slope of a half. So mean square size of those sections is proportional to square root of the genomic distance, so geometric mean between genomic distance and this minimum relaxed section size. So if you invert this relationship, find how S depends on R, you find that the R is proportional to S1/4, or S is proportional to R to the power 4. So on scales bigger than this relaxation strand, the fractal dimensionality of chromatin fractal dimensionality is 4 because it doesn't have time to relax, and it's because of the activity of the cohesions, it gets packed into structures which have fractal dimensionality higher than space dimension. And so we can do a simulation of this. So we could show in here simulations of the polymer without extrusion, and the mean square size of the sections proportional to genomic distance. But if you turn extrusion on, you're getting crossover from this behavior to fractal dimensionality of 4. That's just simulation showing this calculation is consistent with simulation. Any questions on this? This is the main idea of this model. So what we are claiming is that chromatin is packed in our nucleus with fractal dimensionality of 4. So just to summarize, if you look at distance between two loci, if the chemical or genomic distance between them is less than this S sub R, fractal dimensionality is going to be closer to 2. If genomic distance bigger than between them is bigger than this relaxation blob, the fractal dimensionality is 4, and the mean square size is proportional to square root of genomic distance. And so here we show this result from simulations. We see that the distance between two to loci initially behaves as random walk with dimensionality of 2, but it crosses over to dimensionality of 4, and the longer you wait, the longer the section gets extruded, the further and further this crossover occurs. So you get larger relaxation blobs. The crossover occurs at larger s with time as you look at larger sections with loci I bigger than. So this crossover grows, but you still get this dependence of dimensionality of 4. What does it tell you about contact probability? If you don't have extrusion, we observe in simulations, as we expect from the theory, the log plot of contact probability is a function of dynamic distance is proportional to exponent minus 3/2, saying the dimensionality is 2. If you turn extrusion on, you get crossover at this distance crossover S sub r to the slope of minus 3/4. This is what I observe in simulations, as we predict from the theory, and in agreement with experiments. And so that gives us an estimate of this crossover location, which is about 35 kilobase pairs. So this relaxation blob that we observe here in the simulations correspond in experiments to about 35 kilobase pairs. And it's consistent with the parameters we put into the model. So we can do now multi-cohesion contact simulations, and we expect 3 regimes. On scale smaller than the relaxation blob, you just have normal behavior like you have in the relaxed polymer. On scales where activity is important, above relaxation blob, but smaller than this size of TEDs, largest loops, you expect cross behavior with the dimensionality of 4. On larger scale is much bigger than the size of TEDs, you cross over back to random structures or something else, depending on where you have entanglements about. And so that's what's observed in our simulations. We see this crossover from minus 3/2 to minus 0.75 and back to 1.5. And it occurs at the location of the TED that we put in, which in this case, we were simulating for all 3 values, processivity, separation, and size between stop size, which is size of topology associated domains, all the way about 200 bits, which corresponds to a couple hundred kilobase pairs. So we can kind of simulate and reproduce pictures similar to what we observe in the high C experiments. We don't have any details of this off-diagonal domains because we don't have which called territories. We don't have them because we don't have any other interactions. With just all the bits are identical. There is no special interaction that would induce this off-diagonal rectangles. That's why we don't see them in simulation. That's beyond the current model. We just try to explain all the squares log the diagonal. So to compare this in more details, these regions with crossover minus 3/2, 1/4, and 3/2, we can look at the micro-C experiments that seems to see similar behavior, and our theory here is shown, which puts a crossover between the 3 domains into such something smooth, as shown here in the bluish or violet line, and the green line is the data from experiments. And if you look at the slopes or exponents, the simple scaling model has this step signs. If you do a smooth crossover, you get this smooth line from the theory. And the 2 greenish light and dark greenish line are 2 data from 2 different cells experimental from micro-C. So we capture the features with this region, important region from tens of kilobase pairs to hundreds of kilobase pairs, where you have this very high density packing. To understand what happens on small scales, regions up to 30 kilobase pairs have time to relax. Again, dimensionality is close to 2. On intermediate length scales, they get strongly compacted, and dimensionality now goes higher than space dimension. They're about 4, which is what observed in simulation, and experiment, and in the model. And then on larger length scales, these sections of higher compactions form random walk, and they go back to something which had dimensionality lower than space dimension. So first question I can ask is how can four-dimensional object fit into three-dimensional space? And to explain that, is there enough space in three-dimensional for this four-dimensional chromatin? So we can calculate the volume fraction of chromatin inside the volume. You divide the mass or genomic distance by the pervaded volume, and this goes as distance to the power of 1 minus 3 over d. So if d is less than 3, this volume fraction decreases with s, and then what happens up to 30 kilobase pairs. So you can plot the volume fractions on those sections. The volume fraction on log scale decreases with slope of -0.5, up till it gets to the size of this crossover 30 kilobase pairs, and then you go to fractal dimensionality higher than 4, higher than 3, which is 4, and it becomes more and more dense, and it grows with the slope of 1/4. But then it goes all the way up to the processivity or size of TED or something, and then it goes back to random walk, and it continues going down with slope of -0.5. So this is how there is enough space inside the chromatin because of this initial decay creates another empty space, and the volume fraction doesn't go above being too dense. Next, we want to explain why the cell wants to perform all this extra work. And the reason it does is because it increases the overlap of the sections. It wants to have a control between promoter and enhancer. You want to have a very high contact and overlap. You want to avoid overlap between off-target sections. So we can define overlap parameter, which is the reciprocal of volume fraction. So if volume fraction decreases, the overlap parameter, which is number of sections of a given S that overlap with each other, increases initially. Starting from 1 goes to 2, 4, 6. Then, but it doesn't get to entanglement. You need to get about 10 to get entangled, 10 sections to be entangled. Before it gets there, you get this active extrusion that cuts off this growth and creates something which has higher volume fraction or low overlap parameter. So overlap parameter of a section with itself decreases as you go larger and larger sections all the way to the TED size. And then it increases again, but that beyond that, you don't really care too much because you're doing all of your epigenetic regulation on the scales here within a TED, because this is the size of genes. Or distance between the promoter and the enhancer or silencer.
And just giving you a couple-minute warning, we have about 10 minutes left till the end.
Okay, sounds good. Thank you very much. So I'm just going to summarize. So this idea here is regulatory elements and gene promoters are closer in space, and you can calculate this probability of intra-TED contact, and it gets up to 90%. And so you mostly have contact within your TED and no contact with between sampling TEDs. That avoids crosstalk between different TEDs. I'm going to skip this mean square displacement story. Just conclude that location of TEDs and compartments are determined upon cell differentiations and lead to high C contact probability map. And epigenetic regulation is controlled by contact probability of promoters and regulatory elements. And the way it's organized, it allows to have very high probability of contact between promoters and enhancers or silencers and you're getting very special structure. And our model also explains this non-monotonic probability of contact between TED regulatory elements like TEDs. These corners here, which are the peaks here, have non-monotonic dependence on distance, on the size of TEDs, and also we predict the non-monotonic dependence along the edges. So this allows us to understand the structure and allow to engineer, rearrange the structure so you can modify epigenetic regulations in some cases for diseases. So this is what I call the loop of lights. You have the active loop extrusion compacts chromatin on the scales and reduces suppresses entanglements and allows this intra-TED contact with high frequency and reduces off-target contact with very, very low probability, that's way below to 10%. And the few remaining contacts are remaining within these compartments. So all this work was done by Brian Chen, and we have 2 publications in PNAS 2 years ago and last year. And I want to acknowledge support from NSF, EFRI, this was our BME center here, Manet, NIH, and some support from our group in Japan. And thank you much for your attention.